Okay, so hi everyone. I'm Hannelore, or you can call me Hannah, and I'm a PhD student from TU Leuven in Belgium. And in my PhD, I'm researching how predicted protein structures can help us better understand phages. Now, before we get started, I also want to thank the organizers for hosting such a nice conference day and for giving me the opportunity to speak. Now, let's get started. Why am I so excited about phages, and why do I think you should also be? Well, I would dare to say it's a phage's world that we are living in. Phages are the viruses of bacteria, and as such, they are the most abundant creatures on this earth. There's an estimated amount of 10 to the 31st phages on this planet right now. And as viruses of bacteria, they're also true master manipulators of microbial life. They are impacting processes from carbon cycling in the oceans to your own gut microbiome. And this potential to manipulate microbial life at large scale, but also at very specific scales, manipulating all different sorts of cellular processes, actually makes them very interesting from an application potential. Indeed, phages and their proteins have been very important for molecular biology discoveries, but also medicine and biotechnology. In recent years, for instance, phage therapy is really booming, which is the therapeutic use of phages to combat multi-drug resistant bacterial infections. Moreover, many key biotechnological techniques, such as gateway cloning, several restriction enzymes, actually come from our research from phages. Now, despite all of these interests you could have in studying phages, there is still a huge amount of unknowns. Over 70% of all proteins from phages currently have no known function. So there's a huge viral dark matter, and this mainly stems from the fact that actually phages harbor an immense genetic diversity. For most phage proteins, there's simply nothing similar in our databases. But how is the about 30% of phage proteins that is annotated today getting annotated outside of experimental methods? Well, today, if you use phage-specific annotation tools, you're most likely either using sequence information, such as with Verroca, genome synteny information, such as in tools like Fintany, and more recently, also predicted protein structures, such as implemented in Fold. Which brings me to one of the key questions of my PhD research. How much more can we learn about phages when we are using predicted protein structures? Now, while George Bourras was developing Fold, I was simultaneously doing the same. I was predicting protein structures and then using Fold-seq to actually annotate those proteins. However, George and I were making some different design choices. We were looking in different structure databases. We were using different methods to extract hits from Fold-seq, which made us wonder how much of your design choices actually influence the annotations you end up with. And so we decided to join forces to investigate this, and the results is, of course, what I'm presenting today. To investigate this, I made a data set consisting of all the phages that can infect the Pseudomonas bacteria, simply because that's the bacteria we work with the most in our lab, so we have the best expertise to evaluate our annotations. I extracted all their proteins from NCBI protein, which is about 120,000. 71% of those proteins are currently labeled hypothetical protein or phage protein, and we can thus consider unannotated. However, that's still quite a big data set. And so I reduced this data set by removing any duplicate entries and some size filtering, which left me with 38,000 proteins. But there's still quite some homologs in there. And clustering with MM62 at 90% sequence identity and 80% coverage actually showed that there are about 10,500 groups in there. And in these groups, we most likely have homologs, and we can just transfer annotations. So for these 10,500 proteins, my first step was, of course, generating structures as an important prerequisite for structure-informed function annotation. Fortunately, ColaFold can actually predict accurate structure prediction for the majority of this data set, as you can see from this PLDDT plot. PLDDT is a local confidence score of your protein structure prediction, and in general, a PLDDT score over 70, which is the blue bars on my plot, are considered to be accurate, trustworthy structure predictions. And as you can see, the median PLDDT in my dataset was 79, so good structures, we can move forward. This was somewhat surprising, because actually during the structure prediction, we could only build very shallow MSAs. For most of my data set, MSAs contained less than 30 sequences, and the median MSA depth was still below 100 sequences at 99. But nonetheless, we had accurate structure predictions, and so we could actually move forward with these structures and use them for function annotation. Now, if you're thinking about structure prediction with these structure-based annotation with these predicted structures and fold-seek, I guess the most simple method you could use was actually just search a structure database with fold-seek, take the first best hit you get by fold-seek, and just transfer the annotation. For our data set, if we search against the PDB database, we can annotate about 8,000 proteins in this manner. This is also about the same amount of proteins that actually shows any significant structural similarity to the PDB database. When searching against the AlphaFold database, we have an entirely different picture. There, almost 25,000 proteins actually display significant structural similarity. However, if we just look at the best hit to annotate our protein, we can only annotate about 5,000 proteins because most of our proteins are actually most similar to other unannotated proteins, so that's not really helping us. Now, of course, most annotation algorithms actually already think of this and don't use a best hit algorithm, but a best annotated hit algorithm, where you cycle through your hits until you find the first hit that is actually annotated. Doing this indeed improved our annotation rates. For the AlphaFold database, we go from about 5,000 to 15,000 annotated proteins, which is also very similar to the annotation rates that you could get with fold. Now, in this plot, you can also see that there is some other differences with fold. Fold is actually searching phage-specific structure databases, which is resulting in a much larger fraction of our proteins displaying structural similarity to the database. So to conclude, with a best annotated hit algorithm and searching against phage-specific databases, we could annotate 17,000 of our unannotated proteins, which is about 4,500 protein clusters. However, we thought we could take it up a bit further and improve our annotation. How we were doing this was actually considering complementarity. A first complementarity we looked at was at the information in your structure databases and at additional information in protein annotation sources such as UniProt. How are we doing this? Well, for each protein that we are matching in a PDB or AlphaFold database, we actually link back to the UniProt and then fetch the protein annotation information from UniProt. Doing this specifically for the AlphaFold database, again, drastically improved our annotation rates, now from 15,000 annotated proteins to 20,000 annotated proteins, meaning we are surpassing the annotation levels we had with fold, for which no such matching was done because the database identifier conversion is a lot more difficult in that case. Finally, there's also complementarity between these different structure databases because they are each representing different parts of the protein structure universe. And we can clearly see this for the best annotated hit method with UniProt. While a large fraction of our proteins gets annotated by multiple databases, there are actually almost 6,000 proteins that can only get uniquely identified by searching one of those databases. So if you actually search against these multiple databases and then combine your results, you can again improve your annotation rates. But what kind of annotations are we actually ending up with? Well, let's have a look at some of the most common annotations. Looking at this table, I identified the issue that some of my annotations are still not super informative. In my top 20 annotations, I have 3 annotations that refer back to domains of unknown functions, or DUFs. And while this is already better than our hypothetical protein label, it is still not as informative as, for instance, a phage-tail protein annotation. Looking at our entire data set, we identify about 20% of all our annotations, or 10% of all proteins, to be annotated with these low informative annotations. However, if you find your annotation algorithm to actually use 3 tiers of annotation classification during the annotation process so that you can favor high informative annotations over low informative annotations over no annotations, you can drastically reduce this amount to only 7% of the annotations instead of 20%. And with that, I've already shown you some key highlights of how we think you can optimally design your fold-seek-based workflow when you're annotating phage proteins. So my conclusion, I think we should keep folding phage proteins and keep asking questions about these structures. In doing this in this research, we have annotated 22,000 of the 38,000 unannotated proteins, which shifts the NCBI protein annotation rate from 29% to 47% for our Pseudomonas phages. We've highlighted complementarity as one of the key drivers for high annotation rates. And we've also shown that you can actually improve the usefulness and informativeness of your annotation if you use 3 tiers of annotation classification. Now, on a final note, I want to end by saying that these annotations should only just be the beginning, both as a tale of caution as well as an exciting window of opportunity. As a tale of caution, because in the end, all we are doing is transferring annotations on the basis of similarity. And while these are very valid starting hypotheses, they should be that, starting hypotheses, and not a ground truth. However, I also see this data set as a really cool window of opportunity, where with the phage field, we can start diving into this data set and hopefully identify some new cool biology or potential proteins for applications. With that, I'm left to thank all of the people involved, my host labs, Computational Systems Biology Group and Lab of Gene Technology at KU Leuven, as well as the wonderful FAME Lab in Australia, where I had the pleasure to work with George, Susie, and Rob on all of their phage annotation tools. And if you want to know more, I'll be here. I also have a poster in the function session on Monday, so happy to talk more about it. Thank you.
Thank you, Hannah. We have time for one question, if anyone. Okay. Oh.
Thank you. I have 3 questions. Yeah, no, just one question. So how do you do the clustering? Because you show that you do the sequence cluster and then the structure cluster. So how do you combine them together and do the cluster?
So what I've shown here is just a sequence-based clustering. We did structure-based clustering, but I haven't shown it here. But the structure-based clustering we did on all of our fold-seq hits, because I think when you're just using one hit to actually annotate it, you're losing a lot of information. Fold-seq is generally giving you thousands of hits. So we actually clustered those hits based on structure to actually see if the annotation you end up with is actually from the biggest structure group, which generally it is, but yeah. I haven't shown this data, but I can talk more about it later.
Yeah. So you didn't involve the sequence alignment into the structure alignment to do the clustering?
No, so it's entirely separate. So the sequence clustering was just to reduce the data set size, and the structure clustering is when we are looking at all the fold-seq hits. So we're never combining them. It's either one or the other.
Yes. Thank you.
Thank you.
Okay, thank you. Thank you very much, Hannah.